Welcome to our third webinar. Today we will discuss the molecular biology experiments that we have conducted during the Long Night of Science. More specifically, we brought urban potato growers to join us at the museum to extract the DNA of their potato plants. Yes, next slide, please. To give a little bit of an overview of what this webinar series is about, this is the third of hopefully 8 webinars. During the first one, we discussed what is citizen science and what does it have to do with agriculture and the food industry. And we also had a look at the Open Soil Atlas project as well as the PataFEST project. During the second webinar, we also discussed how we are developing a biofertilizer made from mussel shells. And some of you have already tried it or are considering of trying it. We can also ask or have a quick Q&A on this later, but if you want more information, feel free to visit the webinars. But yeah, today we're here to talk about the experiments, and for this I will give a quick overview of what we did. Next slide, thank you. So you see there was a lot of lab coats, lots of gloves, and of course dinosaurs. We had people join us to, as I said before, bring in their own potato leaves, and then using equipment that was borrowed to us from the lab of the Natural History Museum, we allowed people to extract the potato DNA by themselves. And this is basically what it looked like. And well, the question was also, why are we doing this in the first place? Turns out if you look at the DNA, you can figure out what kind of diseases your plant might be resistant to. So right now there's 266 known pathogens that affect potato health, plant growth, or they can also have, let's say, aesthetic issues with the plant itself. And we wanted to see, yeah, basically which of those are resistant. Next slide, please. I also see there's a slight design issue, but that's fine. So yeah, basically some potato varieties have evolved to be resistant, and we wanted to see which of the ones or which of the potatoes that the participants have grown show resistant to certain diseases. And this is in a nutshell what we did. So some of you participated and might remember what we did. We started with extracting the leaves and putting them into, thank you, thank you, Betty, and putting them into very small tubes. Then we added a solution as well as heat to break the cell walls. And then we ran the mixture through several filters so that by the end of it, we ended up with a DNA sample. So that was it, just as a nutshell, just to brief you. And Betty will now go through the different steps and also share with you the PCR results.
Thank you very much. Yes, so now we're going to go a little bit into the results, what they mean, and the further analysis that we also performed on your samples. So the first thing that we did is, and this we already did at the museum on the Long Night of Science, is to visualize the DNA. And to do that, we ran a gel, agarose gel, and we just put a little sample of the DNA into the gel. And this DNA was stained with, it's a stain that intercalates DNA. So whatever glows in this picture is DNA. The number, well, actually, yeah, exactly. The numbers are the samples. And then what we see is, in some cases, a smear of DNA, actually in most cases, and then a big band. So for example, if we look at 16, that is down, you can see a really big blob of DNA. And this is very good quality DNA. because we only have big fragments. If we look at, for example, number 18, we see that it's a smear and the bottom is a little less bright than in 16 and 17. And that means that the DNA, yes, we have good quality DNA that is also big, but we also have a lot of DNA that is small. So it's fragmented, it's not intact. It's not the best quality of DNA that we could have. But the good news is that in all cases, we do have at the bottom a bright blob. Maybe in the case of 2, 3, and 4 is less than in others, but as you can see, as you will be able to see in the next slides, it was enough to perform the experiments. So at the end, we can say we successfully extracted DNA from the potato leaves, and in some cases, even from potato roots, because when we asked our participants to bring their potato leaves, some people accidentally brought in their potatoes instead of the leaves. And we decided, okay, it doesn't matter, let's experiment a little bit. And we decided to extract DNA from the roots, even though we have never tested this protocol in roots. But it actually did work, as you can see in sample number 7 and 8. We managed to also extract DNA from the roots of the potatoes. I will move on to the next slide if I can figure out, yes. So now, how can we find out if the potato is resistant? For this, we need to do a PCR. So now that we already extracted the DNA, we can do other analysis on this DNA. And maybe you have already heard PCR because during the pandemic, it became kind of a normal word to say for everyone when we needed to take a test to do a COVID test. It was either a rapid test or a PCR. But do we know what it really means? So I'm going to explain you what this PCR means and how it works. So the PCR stands for polymerase chain reaction. Polymerase is an enzyme that is actually inside every single living organism on Earth. Because DNA replication is so basic, everyone has this. Bacteria, viruses, plants, animals, any single organism on Earth has this because we all need to replicate our DNA. So it's one of the things that evolutionarily only happened once, and that's it. So based on this thing that already happens inside our bodies and all other organisms, some scientists created a protocol to help us obtain or recognize pieces of DNA that we know inside a genome. In this case, the potato genome. So what we need is first a DNA template. And in this case, a template means the DNA that we extracted from our potatoes. Then we need primers. And primers, as you can see here, are small pieces of DNA that correspond to the gene that we are looking for. In this case, the gene that gives the potato resistance to certain pathogens. Then we need nucleotides, because nucleotides are the parts of the DNA that we need, in this case, to complete the whole sequence of the genome. We need polymerase, that is the enzyme that is going to perform this action. We need a PCR buffer, that is just some substances that allow this polymerase to survive. Otherwise, it can, if it's not the right pH or the right temperature, this polymerase can degrade and not to do its job properly. And then we need to put it in a thermocycler. And the thermocycler is a machine that will go at different temperatures as we want to do it. And you will see why we need the different temperatures. The first thing that happens is denaturalization. We have DNA that is a double strand of DNA. So it's two strands that are together. And the first step in our PCR is to apply heat, 25 to 26 degrees. And what will happen to this DNA? Instead of standing together, it will separate. And we will have two separate strands of DNA. Then we have the annealing step, and that one is performed at 68 degrees. So this machine will now decrease the temperature and go to 68 degrees. And what happens in this step is our primers will stick to each side of the DNA. And we have two primers, one for the DNA that goes in one direction, so one strand, and the other for the other strand. They will stick. If they find their match, they will stick. Then we have the action of the polymerase. What the polymerase does is it reads the information from the template, so the original strand, in this case is the red one, and it continues to do basically the pink part. So it knows that the sequence because of the template, and it will take every nucleotide and it will attach it to create the whole sequence of the gene that we are looking for. And at the end, it will do this several times. And the elongation is then done at 72 degrees, and that's just the completion of the sequence. At the end, it's done many, many, many times. And because each time we have more and more template, because every time the polymerase finishes, we have more DNA that corresponds to this sequence. So in the next cycle, we will have even more and more and more. So that at the end, we are not going to see anymore. I'm gonna go back to the image of our DNA. This is the entire DNA of the potato, but we don't wanna see this. We only want to see the gene that is giving resistance to the pathogens. And when we do this, now it happens so much that most of the DNA that we have now in our little tube is the DNA that we want to see. And the rest is neglectable because we have so much of this specific DNA. And the result is, but first of all, I wanted to tell you which pathogens we tested for. So as you heard before from Leo, we know at least 264 different pathogens that attack potato plants. And in this case, we only tested for 2 of these pathogens. Number 1 is the potato virus Y, short PVY. And this is a virus that is transmitted by aphids. Aphids are insects. And the insects get infected from this virus, but they don't get sick. They simply pass it on to the potato. They get infected by eating plants that have been infected. And then when it moves to eat to a healthy plant, it immediately passes on the virus to the healthy plant. And we also tested for another pathogen that is Globodera pallida. And this is a worm, a nematode, that is known to infect many plants, but especially potato plants. And it is one of the most important pathogens that affects economically potato growers around the world. So we tested these 2 pathogens. We wanted to know the potatoes that we have, if they are resistant to these 2 pathogens. And these are our results. In this case, the bright blob is the presence of the gene that gives resistance to the potato. So if you have a band, it means the potato is resistant to the pathogen. If there is no band, for example, here in 7 and 8 and 11, that means these potatoes are not resistant to the pathogen. And these numbers are the same as in the first gel image. The first image on the left side is the HC, which is the worm. And on the right side, you have the same thing, but with the virus. And in this case, coincidentally, the same potatoes are resistant to both, and the same potatoes are also not resistant to both. So these are the results that we have for your set of potatoes in this case. Basically, those are the results, but I can imagine that you have a few questions. So I'm gonna allow you to ask some if you want.
Maybe I can start with one. Yes.
Uli has a question first,
okay,
but she's muted. Uli, did you want to ask?
Yes. I wanted to ask, what does resistance actually mean? If then there is a potato virus coming and then nothing will happen, or only a little bit, or?
It actually means nothing will happen, but it also depends on the kind of pathogen that we have. So because we have so many, and there have been already many studies that have tried to do collection or like a database of different potato kinds, and they have tested almost every single pathogen that we have, and in different cases it means different things. So for different pathogens, the resistance might mean a little bit different, but I can look specifically for these two what it means, and I can tell you more details later.
Mm-hmm. Okay.
And Leo, you also had a question.
Yes, I was wondering, can you go back to the DNA extraction results? Because also based on this, I realized that 3A might also belong to Michael, right? Because to me also 1 and 12 look similar, but I also don't have the background, and I was wondering if that's, for example, the same plant it shows on the DNA extraction results.
We cannot really say much about that. We can also, so the gel, even though it is a little bit of quantitative, so you can see, for example, if you have a big band, you can infer that it's a lot of DNA, and if you have a small band, you can infer it's a little bit of DNA. So it's semi-quantitative, but it's not really quantitative in the sense we cannot really know how much DNA there is. And we also cannot really say it's also not qualitative because it doesn't tell us anything about the potato. So we can only see the results from the extraction. We can only infer that the extraction was successful.
Okay, so based on this, we could also see that the extraction was done very similarly. For example, 16 and 17 was done by the lab, and the fact that it looks so clean is an indicator of that. Okay, thank you.
Exactly. So as Leo said, number 16 and 17 are samples that we extracted in the lab. And of course, it's a different setting. The lab is always much more clean, and we did it alone without people walking around. So the result shows that it's a much better result. But in this case, it didn't, as we can see then by the results of the PCR, it didn't matter that much because at the end we were able to do PCR on the samples.
Mm-hmm. And what about this 7 and 8? Are these roots, or is this was it also leaves?
7 and 8 are roots.
Yes, and that's strange that you can't identify this resistance in the roots, no?
But so we also have sample number 11 that is the same potato as the ones from the root. And if you look at the result, number 11 is also negative.
Yes.
So we cannot identify, so it's really the potato. It's not just the roots, it's that potato specifically that it's not resistant to these two pathogens.
Uh-huh. Hmm.
Are there any other questions based on the PCR results or the DNA extraction? I mean, I can also, or we can also mention that we did not add a positive control to this. So Betty, I guess this is something that is usually done as well, right? To have a negative and a positive control to double check.
Exactly. So in this case, we knew that the plant innovator was resistant. And do you know which sample was that, Leo?
If you go back to the table, I should be able to tell you. Oh yeah, Maria. So oh yeah, 4 and 5 probably not.
So number 4 and 5. Sorry, did you want to still check? Ah, sorry.
No, then it's 4 and 5 and 14 and 15 as well, because
Okay.
those were attendance from the long night of science.
So 14, exactly. So we had positive controls. That means we knew for sure that these samples are resistant to both pathogens. And that was our control to understand that our primers are working.
Mm-hmm. Yeah. And another question, is it normal that most potatoes are resistant against these pathogens?
I think it was a coincidence from the different potatoes that were brought by our participants. We would have to have a lot more potatoes, different kinds of potatoes to see if this is really like a majority thing or not. But there are some studies out there that really tested many, many different kinds of potatoes, and it doesn't seem to be a majority. It's maybe like for some pathogens, yes. We have to also consider that these are very common pathogens, these two that we chose. And that means that they have attacked a lot of potatoes, which also gives a lot of pressure to the potato plants to evolve to be resistant to them.
Mm-hmm.
So if they are, if there is this, it's called evolutionary pressure, then the potatoes most likely evolve to have this resistance.
Mm-hmm.
And it works better. So if it's a pathogen that is not deadly, but it makes them sick, but not sick enough to die immediately, then it gives them more opportunity to evolve. If it would be a pathogen that immediately kills the plant, then they probably will not become resistant because they die too soon and they don't have the opportunity to, they don't have the time for their DNA to evolve.
Mm-hmm. Well, thanks.
Any more questions? I think it's not the case, Leo.
Yeah, it looks like it. Then I thank you all for attending. I would like to invite you to also have a look at the podcast, which is Potato Talks. We actually, oh no, I was about to say we did post an episode on the Long Night of Science, but instead we have several social media clips that you can follow. So not a directly an episode on this, but if you go to the next slide, we have a Menti where we would ask for all of you to fill it in. And again, in the meantime, feel free to ask more questions about the about anything relating to PataFEST really. But if you find the time to answer this, it takes 30 seconds just so that we get a rough overview of whether the webinar was useful for you or not. That would be great.